Okay, let's talk cell mitosis. So cell mitosis happens when like an organism needs to grow or to heal, or if it needs to like asexually reproduce, like bacteria and stuff. There are hold on. Most of the whole cell cycle of mitosis is actually just interphase. G1, S, G2 are all interphase, and mitosis is this just like last itty bitty slither of it. So in G1, the cell is growing, getting ready for DNA replication. In S, the cell is replicating its DNA. And in G2, it's growing even more, replicating specific organelles, microtubules are made, etc. So the way I think about it is that the G's in G1 and G2 are like standing for growth, right? Because the cell is growing. Afterwards, there's mitosis. So let's go through the phases of mitosis. First, there's prophase, where chromosomes are condensed, the nucleus is devolving, centrosomes move to opposite sides, and spindle fibers begin to form. Right, so this is our nucleus. It's beginning to break apart. Our chromosomes right over here are pairing up into sister chromatids. Yeah, that's about it. The next is metaphase. The nucleus is fully dissolved at this point. The chromosomes are lined up at the center like this, and the spindle fibers that are from the centrosomes are attached to the chromosomes like this, right? And in anaphase, they are teared apart, right? And they're pulled so each sister chromatid goes to the left or to the right. Afterwards happens telophase and cytokinesis, where the nuclear membrane reforms, the centrosomes revert to centrioles, and the chromosomes start to unwind back to their own like original DNA shape. And with cytokinesis, the membrane is actually pinched in like animal cells or whatever to have two separate somatic cells. Anything that isn't made through meiosis is a somatic cell, which basically means that they're like the same. The S is for same. In mitosis in general, there are 3 checkpoints. At the end of G1, there is a check if the cell is large enough, has enough resources, and if the DNA is damaged. If it doesn't pass this one, it tries to repair itself, or it goes straight to G0, which is like retirement, right? It stops replicating any more cells. Then there's another one at G2, where it asks itself again, is the cell large enough, or if the DNA is properly copied. If not, it will try to fix it. Then with the last one at metaphase, are the sister chromatids lined up correctly? Are the spindle fibers attached right? If the answer to any of these questions is no, the cell will try to fix the problem or commit apoptosis, which is like cell suicide. So in these 3 or 4, I guess, steps of the cell cycle, there's these things called proto-oncogenes and tumor suppressor genes. And those are positive control and negative control, where proto-oncogene is positive control and negative control is tumor suppressor gene. So a positive control is basically like pressing on the gas, saying like, okay, keep on going. Keep the cell cycle going. Everything's looking good so far. A tumor suppressor gene is basically saying like, hey, step on the brakes. We need to slow down. We need to fix what's going on here. Like my DNA isn't replicated correctly. So let's go through positive and negative control more in depth. Negative control is done by 3 proteins: P53, which does cell suicide; P21, which disrupts cyclin CDK; and RB, which disrupts other necessary proteins, right? So these ones specifically are basically like, I'd say like cutting the brakes or like pressing on the gas right now. Cutting the gas or pressing on the brakes to stop, the cell from going in any process that it's not ready for yet. Wow, apoptosis is just blowing up the car. Positive control is done through 4 cyclins, which increase concentration at different cell cycle stages. Each cyclin binds with a CDK, right? The same CDKs from P21, which move the cell through a particular part of the cell cycle. Right, so Each stage it checks like, hey, is this working correctly? If so, it presses on the gas with one of these 4 cyclins. Now before we go through meiosis right over here, I'm going to talk about some really quick vocab. So if you have 2 cells together like this, you have a diploid chromosome. Okay, it is combined with a centromere in the middle, and the 2 pieces together are sister chromatids, right? Because they're the exact same, they're like sisters. You'd classify this as 2N. Now if you had 2 exact same chromosomes like this lined up together, you would have a homologous chromosome pair. If they're on top of each other, then it's a tetrad. Now if you have this diploid thing split in half, like we did with anaphase, you would have a haploid chromosome, right? Or just an N. Now let's talk through meiosis. There is, I think 8 phases or something, double of whatever mitosis had. Let's start with prophase 1. The chromosomes are duplicated already, hugging together like this. The nuclear membrane is dissolving, asters form, and the crossing over happens, which is where the homologous pairs will share bits and pieces with other parts of them. So like, let's say this top left half or top right half swaps with this top right half, like a little chunk. And then that increases genetic diversity, so you don't have like a 25% chance of looking exactly like your dad or exactly like your mom. These are small itty bitty things that make you more unique. Okay. Then there's metaphase. The homologous pairs are lined up just like this. There's random orientation where some of your chromosomes from mom go to the left and some of them go to the right, and the same thing for dad. Then with anaphase, the spindle fibers shorten, homologous pairs move apart, right? That's like almost regular mitosis anaphase. Then we have telophase 1, which is basically the same thing as regular telophase, except everything is diploid instead of haploid. Now, I'm going to be focusing on one of the 2 cells, but keep in mind, this process happens for both of these 2 cells. So in prophase 2, there's no duplicating that happens prior, and it's basically the same as prophase 1, other than half of the number of chromosomes being present. Right, so your nuclear membrane is still dissolving. Actually, no crossing over happens because it's not possible. But the asters form again, same thing. Metaphase 2 is basically the mitosis metaphase, right? You're just like lining everything up the way it needs to be. Then anaphase 2, it's basically the mitosis anaphase. It rips everything apart again in half. And then we have telophase 2, which is basically mitosis telophase, and that creates 2 gametes of 4 total, right? So this cell makes 2 gametes, and this cell makes 2 gametes. So in total, there's 4 gametes made. So what is a gamete? A gamete is a reproductive sex cell that's found in animals, like with sperm or eggs in humans, or like pollen in plants, right? 2 gametes combined, if they're the right kind, create a zygote, right? It looks something like this, and this is what allows for human life to be created. There's a few things that can go wrong with meiosis. There's a non-disjunction when homologous pairs fail to split apart during metaphase 1. So right here, it would be like this one sticks to this one right here. So there's 1, 2, 3, 4, and 1, 2 here. Trisomy is a specific kind of non-disjunction where there's 3 chromosomes in the gamete cell instead of, the regular, or there's 3 chromosomes in a zygote instead of the regular 2. So you'd have 3 X chromosomes or something like that. Translocation is an error in crossing over when genes are swapped with the wrong chromosomes. So think like a big chromosome like chromosome 3 swapping with a really small chromosome like chromosome Y. Okay, that would not be good for the cell and any chances it has at becoming a fully functioning human. Now, there's when DNA is being replicated in synthesis, there are some errors that can occur, right? So one error, for example, is deletion, where one base is removed and everything gets moved backwards like one piece. And then at the same time, there's also insertion, where one base is added, moving everything forward one piece, right? And this can really affect the creation of proteins. It can basically lead a cell to being completely pointless or even becoming cancerous if the problem is not corrected. Other than that, though, that's just about everything that you need to know for mitosis and meiosis when it comes to the cell cycle.